excitement, so I'm going to definitely that slightly disrupted our conference. But can you take a second and just again reiterate the awesome news and the grant you got? Oh, my.
For the other one? Okay. Sorry, yeah. I'm still sleepy a little bit. So, yeah, we received like the notification of award of our R1 is a 5 years grant from Child Health and Development, the Institute, NIH Institute of Child Health and Development. And this grant is about CHAMP1 and brain development. So we are going to study, and this grant basically will fuel the research for CHAMP1 and its role in brain development. So, and we are going to use also this money to work on the therapeutics with the Commons Lab. So that's amazing. Thank you.
Congratulations, and a equally big congratulations to our community that he gets to continue and expand what he was already doing. And it's amazing news for us, and probably one, if not the biggest news. So I just didn't want that to be overlooked, because I don't think our community understands the significance of it. And we're fortunate and grateful to have you and everyone else at MUSC here. And honestly, we just can't say enough good things about all you guys. Andy, I spoke to you yesterday, and I didn't know what we were going to walk into Sunday, if we're going to have as many people. I knew it'd be a lot more casual, but from an outside perspective, and I think, you know, it's been 5 years since our last sit-down face-to-face conversation. What did you see yesterday, where we are as a community, and maybe some of the things that we're missing, or maybe some of the challenges, and sort of what you think we need to have in terms of our ducks in a row, so when we get from preclinical to clinical, there's no setbacks?
So, hey, my name's Andy. I was joking around with some of the research team last night at dinner. I think I may be the only industry person here. So I work for a large, hopefully this makes me sound smarter. I work for a large gene therapy company, and got to know Jeff 5 years ago on some nonprofit efforts we make to make gene therapies accessible for groups like yours that, you know, are likely not investable, right? And that doesn't mean it's not possible, it just means it's not investable, so they get ignored and left behind. I told Jeff this, you know, we tend to look at kind of the checklist of stuff of if somebody's ready, and I'm a specialist player in all this, right? So I kind of hope that groups like yours never need groups like ours, because if you have to get to a gene therapy, it means a lot more simple classes of therapeutics don't work, right? If you could do it with a pill, do it with a pill. If you could do it with an ASO, do it with an ASO. If you can't, then that's what we exist to try to help with. I don't know how it feels for the patients and the parents here, but it's incredible amounts of progress. It's got to be tough to feel like you're just bashing your head against the wall. But you're not. There's real progress here, and I think there's something here that's a little bit out of my comprehension even, but between what we heard from the clinical presentation and the iPSC models yesterday, there's really something interesting there, and to get an R01 grant from the NIH is not nothing either. That's typically the stage right before there's another class of NIH grant to get a therapy going, right? It's from here to there to therapy. Therapy is clinical trials, and again, I don't want to make everybody, you're dealing with groups who have clinical expertise already. So MUSC has a huge research hospital, St. Jude has a huge research hospital. For me, the only piece that is a little bit confusing for me is seeing just what you were alluding to, the total deletion of the gene is a milder phenotype, but in the cell models, it's not. That suggests that there's multiple different therapies that may be needed to do this, right? And that deeply understanding the nature of the mutation is key to a class of therapies where some of these patients may need a simple answer and some may need a very complex answer. And I will say it's really unusual for the genetic diseases that my group works with that we would see total deletion being the mildest phenotype. And I apologize, I've been trying to reach out to parents and patients here as much as I can to do some nerd translation. So if anybody doesn't see that, or if I'm doing the thing that we should all be conscious of, and that's the most interesting thing I've seen from the last 2 days. But I think other groups have gone much longer with much more complicated or much simpler diseases from a mechanism of action and not gotten nearly as far as you guys have. And I don't know how to be realistic about the challenges, but also celebrate the progress. But Jeff, from the first time we spoke to now, it's pretty incredible. And I think it should be really proud for a group of parents and lay people to have pushed this hard, this far, this fast. I mean, not the least through COVID, like y'all, but that ruined everything for everyone, right, in terms of rare disease research and fundraising. So that's what I'm taking away from this is that dichotomy, that difference between kind of the 3 flavors of genotype and the 3 flavors of severity suggests 3 possible answers to me. But having 3 possible answers, not 5 million, is a hell of a thing. So that's my conclusion.
Thank you. And I think, and this is a parent's perspective, I mean, no one doing this for thank yous, and I think the celebration is when we get to some sort of a treatment, some actionable spot in our community where we see progress. But it is, you know, not very often do I personally take a chance to reflect on the growth. I know we're in the right direction, and it's for people like you and that are in this room. For the remaining researchers that are here, and I don't know if you can or you mind, but are you okay or comfortable with maybe like a 1-minute summary in layman's term for some of the families? I know Daly did an awesome job yesterday of speaking to our community. I think that was very clear. Some of the feedback we got from parents is that was understandable and straightforward. Is there any other project, Dr. Tanaka? Again, I know I've said this 5 times already, but your poise through your poise through the distraction was incredible. But I need to go back and rewatch your video. I may have missed a few points. Is there anything after today, with all your years of experience and expertise in CHAMP1, do you feel like, in addition to the question of is it a dominant negative, what is the most important next step or question that needs to be answered for our community?
Okay, yeah, I've been working on CHAMP1 for more than 10 years, but yes, so every time I join this conference, I think you know much better than me about CHAMP1. So I think, yeah, so I always learn from you. So compared to the previous occasion 2 years ago, I think at that time most of the talks are just a planning stage. But this time, I was impressed that there are many results, many achievements. So I think the advancement of these 2 years is surprising. So I hope that there must be some new finding in to a year or 2 years later. So for the dominant negative stuff, I'm cell biologist. So the approach from different point of view, like we are doing right now, is to keep on going. I think very important. And then so I don't have any clinical point of view, but at cellular level, maybe it's not so interesting for you, but still it's strange for me, so that all the truncated mutant have the similar phenotype, because at cellular level, in my mind, it is divided clearly into two categories. So that actually in the middle of the CHAMP1, there is a small sequence called nuclear localization signal. And then when we expressed these mutants, the truncated mutant without nuclear localization signal cannot localize to the nucleus. So that they just distributed in the cytoplasm. So because most, all those functions of CHAMP1 have to be done in the nucleus, to me, it's completely different. So that, very short variant like JJ cannot localize to the nucleus. So it's very strange for me. So still, all the truncated mutants have the similar phenotype. So this is what I can address at cellular level. So that to me, to discriminate, so that to this question will be the next for my next part. Yeah, next part. Okay, thank you very much.
I'd like to add a comment. I agree about an apparently homogeneous phenotype associated with the truncating variants. But also during this meeting yesterday, I realized that there are significant differences with respect to the pattern or the neurodevelopmental impairment, but also with respect to the physical phenotype and the facial phenotype as well. So I raised the question to myself whether it's not a mosaicism, because the mosaicism for the mutation was to me the first hypothesis explaining these great differences between the same kind of variant truncating. But in considering all these possible interactions with different proteins, I wonder whether the difference could be the kind of interaction. So the quality of the proteins involved in this molecular pathway. But to my experience, based on my experience, direct experience by analyzing directly individual kids, I think that there is a subgroup of patients with a very, very mild clinical presentation, although the genetic defect is apparently homogeneous, a truncating variant. I don't know whether it could depend also on the type of the variant. So we have this record, and thanks to the registry of the disease, we could investigate about this specificity of the variant, maybe. But to me, it's not homogeneous, the phenotype. Tends to be homogeneous.
Steph?
Thanks. Again, I just want to thank everybody for allowing us to join this meeting this weekend. This has been really enlightening and interesting. Coming at this from a different gene group, I just want to comment a little on the dominant negative issue. Looking at this from the outside, obviously the simplest explanation from the clinical phenotyping is that it's probably a dominant negative type effect. But I would also caution that it can be problematic to close off the possibility that that might not be the case. And I think, Alon mentioned one possibility yesterday, which is the entire gene is gone, and there's a way for the other gene to compensate, whereas if both genes are functioning, you don't get that compensation mechanism. So there could be some trivial explanations that could ultimately explain the difference. What I see as a major gap is you need a mouse model that is trying to tap into this. And I don't know if somebody's making that, but that is something I think you ought to think about, trying to introduce a knock-in or 2 or 3 of some human patient mutations into the CHAMP1 gene, so you can really start asking some questions about this. This also can be done, obviously, in cell models, but I think what I saw from, and my lab personally tested the knockout mouse from, you remember Jeff convinced me to get the mice and test them a number of years ago, and we really didn't see anything, which may fit with your idea that you mentioned with the human population, that the full deletion is relatively mild. And while there was excitement about the pre-pulse inhibition effect that was described yesterday, you know, it's a pretty modest effect. And if there's anything I've learned in my 30 years of being a research scientist, is it's really, really hard to study small effects. And it's really hard to tease out a therapeutic using that level of small effect. So if a mouse model could be generated, it could really help both address this question more directly and maybe even produce a better model system where therapeutics and things could be screened and tested for potential effects. Anyway, that's just my 2 cents. I know that there's a lot of talk, and we've talked about this a little bit offline previously as well, but I really think trying to nail this down could be a really important going forward.
Just to ask, because so for myself and for parents, I don't know if anyone grasped the mouse data and the phenotype. I know, and you could comment to this, the knockout mouse is subtle, that you told me that before. There's not much of a phenotype. The mutant model that was printed on yesterday, this is your expertise. Was there anything that stood out? Is it a good translational model? Is it something that
you're putting me on the spot.
Sorry.
I think that phenotype needs to be more robust for that to be something that could really be, you know, something you're going to use as a basis of trying to see a therapeutic effect. That's my personal opinion. Sure. But it doesn't mean that a different mouse model may not be superior. And in fact, you know, again, if you can do a knock-in mutant. So, I mean, for those of you wondering what I'm talking about, you know, using CRISPR type approaches now, you can go in and generate a human patient mutation in the mouse, almost exactly what you would see in the patient. And you know, the cost of that from a commercial company is going to be somewhere in the order of about $25,000 to $35,000. If you make it through an academic institution like mine, you know, I get a big internal discount, so to speak. We can make it for probably about $15K or something like that. It's expensive and it's a big investment, but what we're doing with the MEF2C side of things was we're making a matching mouse mutation for every iPSC line we have. So we have a missense mutant patient iPSC line, and we have a mouse that has that exact same mutation. We have a truncated frame shift mutation, we have a nonsense mutation, meaning a premature stop, and we have a deletion. And so we have human cells and mouse to try to match for all that. Do we need all that? I don't really know in the end. Ours seems to be a little more clear-cut of haploinsufficiency for MEF2C. But even in the MEF2C group, we still have this discussion. You know, for some patients, and I think what you said is right on the money, the therapeutic approach may and likely is going to have to be patient-specific. And it's going to require some tailoring. The other thing that I think is kind of interesting to think about as well is that it is possible that the truncating mutations are interfering with the wild-type copies function. But if you were to target that for degradation, it's still not clear if the one surviving copy, after developmental pressure to upregulate that other allele and have compensatory mechanisms, it may reveal a haploinsufficiency at that stage. And so again, I think it would just be smart to just be agnostic about it and keep an open mind about all the possibilities, which I know you are.
I think, and if this is the way it goes, if we just have to keep failing to find the answer, it's fine. I think we're just going to get as many shots on target as we've been able to. We've thrown a lot of darts, and now we want to be a little more strategic about it. But
right.
Last question with what you said, and I'm mad I had it in my tip my tongue until last comment. I'll get back to you, so I'll think of it in a second. Nate, if you don't mind, and I don't know if anyone has a comment on that. I wanted to ask you, and you guys could all comment on this. I know that we've gotten so much good feedback about partnering with POGs and having them there, and I know that there's a lot both of our communities are going to learn because our genes are constantly coming up in each research presentation. Is it realistic? Is that where the benefit of doing this conference ends, or is it realistic that there's a drug, like I know there's drugs that are beneficial for everybody, you know what I mean? There are things that could help with symptoms, but is that a likely approach to find some therapeutic with another group? Because I know we share a lot of symptoms and our kids have some things that are unique to our two groups, but and then they do have a severe population that wasn't present. A lot of the kids that came were pretty high functioning, but their higher functioning kids seem to be relatively higher functioning than the majority of our kids. So is that a reasonable approach to therapeutics, or are we just getting the knowledge side of the research the scientists are doing?
Are you asking like would a therapeutic that works for POGSI be like translated to CHAMP1? Is that what you mean?
Yeah, well for you yesterday was cool presenting the sort of the joint.
Yeah, I mean, if there's mechanistic overlap between the two conditions, that's like a downstream effect that we can target. I don't see why that wouldn't be a potential approach. It's just difficult without like a model or further preclinical testing whether we can gauge whether it has a pronounced effect or not, or what the potency needs to be for it to really be successful. And that's sort of where we reach a dead end, where we have mechanistic reason for a therapeutic to work based on like differential gene expression or whatever qualifier you want to use, but it's difficult without seeing the benefit of it, whether or not we can recommend it based on whatever side effect profiles or whatever barriers there are for it based on like a lack of beneficial evidence.
For the program that you're developing and sort of in the works, what are the data sets that their clinical picture seems to be a little more well established than ours, but I don't know necessarily if they have the resources for not iPSC lines yet, and think maybe scarcely. So what data sets do you need to pursue sort of when that project comes up and running? Is it seq data or is it?
So the thing that's up and coming that would be looking at both POGSI and CHAMP1, that's from my understanding just published data. So like anything on PubMed, that's literature that's published that examines the relationship. I mean, if there's RNA-seq data that shows a knockout of a gene differentially changes the expression of both CHAMP1 and POGSI, that could be beneficial, but that's kind of a case-to-case situation right now. So I would need to talk to Dr. Mike to see if we can include that as well.
Well, we have some remaining minutes before we have our last